This is the model answer video. This is the essay question 3. Core concepts, key assessment 3, paper 1. In the help me video, we really explained the whole thing really. We said we've got an enzyme. So we've got enzyme, which is phosphatase. Pyrophosphatase. That's our enzyme. And we've got a substrate, which is pyrophosphate. And the first part of this essay, describe and explain why pyrophosphate can only hydrolyze. So pyrophosphatase can only hydrolyze pyrophosphate. So that's our first area. So we're talking about pyrophosphate and the structure of it. So I'm not going to write this as an essay, I'm just going to put some key indicative content. So pyrophosphatase, I'm going to talk about the structure, active site, shapes, being complementary. I'm going to talk about enzyme, substrate, complexes, lock and key, maybe induced, fit. I could talk about activation energy. Basically, how do enzymes work? So your key theory on enzymes working. So that's our basic area there. Then we've got two other areas that we're going to talk about, and we're talking about this inhibitor phosphate. We notice that this phosphate has got a very similar shape to pyrophosphate. So this inhibitor has a similar shape to the substrate. Not the same, similar shape. So it could fit into, let's say this is the active site. We're not told it is, but are we? No. But let's imagine that is. So pyrophosphate fits in there, and phosphate could fit in there as well. If phosphate's in there, then pyrophosphate can't fit in at the same time. So it's blocking it, and so our substrate can't fit in and can't change into the product. So this, our phosphate, we're going to talk about it being a competitive inhibitor, yeah. We're going to say the things like similar shape to the substrate pyrophosphate. We're going to say the shape is complementary to the active site. We're going to talk about, well, it goes in, so it blocks the active site, so it fits in, blocks, prevents the substrate fitting in because it's physically there. We could mention the point about increased levels of concentrations of substrate would negate the effect of the inhibitor. It doesn't particularly ask for that, it's in the mark scheme though. And the third area, we're going to talk about our other inhibitor, which was phenylalanine. Don't worry about these words, you don't need to know them, and they're there, so you're just copying them down. And this is the other type of inhibitor, which is non-competitive inhibitor. When we look at the shape of this, it's a different shape to our substrate. So it's not going to fit in our active site. It's the type of inhibitor that fits in a site away from the active site, but then changes the shape of the enzyme tertiary structure, and so the shape of the active site changes, so the substrate can't bind. So we're going to notice its different shape to our substrate. Different shape. Binds away from the active site. So it binds away from the active site, but still causes a change in the shape of the active site. And now it's a problem. If you change the shape of the active site, substrate can't fit, can't find in it, and it can't have an enzyme substrate complex, and so on. So substrate cannot bind, and so less product being formed. You could mention that the substrate concentration would have no effect on this activity. It's in the mark scheme indicative content. Doesn't particularly ask for it in the question, so you'd be forgiven for not saying that. So all you need to do now is, those are your three areas. You need to think of them quicker than we've explained them. You need to say the theory. It's all absolutely standard theory on enzymes and inhibitors, but you need to talk about these examples and shapes and fits and so on. So once you've recognised what these are, the essay kind of writes itself.